tonight we have 3 panelists that are going to be presenting on the American Burying Beetle. And we all want to welcome you all to this evening's event. We hope that you're enjoying possibly the 2 sponsored cocktails for this evening. The one that is it's called the Bury Me Deep, or the Beetle Juice. It's all associated with tonight's topic surrounding the American Burying Beetle. Or maybe you're also potentially enjoying the St. Louis Zoo sponsored beer, the Bier Kölsch Ale. So I'm going to go ahead and introduce our panelists, but before I do, I want to make sure that everybody's aware. We're going to start off with the overall presentation for tonight's topic, but as the presentation goes on, please feel free to enter your questions into the Q&A box that's located on the bottom of the Zoom panel. And as we get done with the presentation, we're going to do everything that we can to get through as many of the questions as possible as time allows. So I'm going to go ahead and I'm going to introduce our panelists, and then we'll go ahead and get started. So I'm going to start with Bob Merz, who is the Center Director for the Center for American Beetle Conservation. He's been a member of the WildCare Institute with the St. Louis Zoo. We also have Kayla Garcia, who's the Zoological Manager of Invertebrates. And we also have Renée Hazen, who is an invertebrate keeper. So tonight they're going to be your panelists, and I'm going to go ahead and hand it off to them.
Hi, Mike. Thanks for doing this for us. This is great. Okay, so let's get started. American Burying Beetle. So the Center for American Burying Beetles is one of our zoo's 30 field conservation efforts all around the world. And it's one of 7 local ones that we have here. Next slide, please. We get asked a lot of all the animals that you can choose, why beetles? And so to pull back on that like a little bit, and just to give you some frame of reference, I like to look at it this way. One way to look at the animal kingdom is to divide it into vertebrates, and vertebrates are what most people think of when you say name a animal. These are the dogs, the lions, the giraffe, the fish, birds, things like that. Snakes are vertebrates, and so on. So if they have a spine, it's a vertebrate. But invertebrates by far make up the vast majority of the animal kingdom. I mean, we're talking 98% of all the animals don't have spines. So next slide, please. If you were to put that into perspective at the zoo, and if our zoo were to represent the animal kingdom more proportionally, the zoo would be comprised of exhibits that would have names of groups of invertebrates. So like, you know, instead of the birdhouse, we might have the fly house, or you know, and that would display you know, the amazing diversity of fly species all around the world. Or we might have Beetopia, and that would showcase some of the 20,000 species of bees that are out there in the world. Or instead of big cat country, we might have Cephalopod County, USA, which would be one of my favorite places to go. So and then all the representatives from the vertebrates. So that's all the birds, all the hoofstocks, all the primates, all the snakes, all of that stuff would be found in an area the size of our current Bier insectarium, and, you know, and butterfly garden right outside of it. So thinking about that may change the way that we look at the animal kingdom out there. Next slide, please. So beetles, let's just hone that down to beetles, because there's a lot of beetles out there too. And if you think about this, if you line up every species of animal on the planet, every 4th one would be a species of beetle out there. And that is amazing. Like they're all over the place in size and shapes and so on. Next slide, please. And the numbers of these are mind-boggling. Folks estimate that there's between 350,000 and 500,000 different species of beetles in the world. Species we're talking about. Next slide, please. So tonight, with all those options, up to 500,000 of them, we're going to talk about just one of them. And the important thing to know from this slide is that this species of beetle, Nicrophorus americanus, was federally listed as endangered in 1989 by the U.S. When I tell the story, next slide, please. So when I tell this story, I often talk about a body that was found 6 miles away from where we had put the beetles out. And this was a body found by a special investigative unit from St. Louis. Next slide, please. Now here's where it gets a little kind of gruesome, because there was a couple there that had hid this body in an underground bunker that they had constructed in secret, basically. Next slide, please. So what this special investigative team found was that the couple had crudely embalmed the body. And this is where it gets really gross. They had intended kids. Next slide, please. So the big reveal is that the couple was a pair of burying beetles. The body was most likely a bobwhite quail. The children were their offspring, and the special investigators were zoo employees doing their work. And I tell it that way. It's slightly misleading, but it's not. I mean, it's all truthful. But you form images in your head, and those images now allow everyone that's heard that story to be able to recount the life history of this beetle species out there. Next slide, please. So here's what happens. A beetle finds a dead vertebrate. So that's something with a spine. And the ideal size is between 100 grams to 200 grams. They bury this underground in a bunker of sorts, and they embalm the body with secretions that they make, and that preserves the body for at least about 10 to 14 days or so. They lay eggs down there. The eggs hatch in 1 to 3 days, and then both the parents feed their larvae from that body for about 1 to 14 days or so. The larvae then burrow away from the carcass underground, and then they pupate. This takes between 45 and 60 days, when they then crawl back up to the surface and they start the whole process all over again. Next slide, please. So there are these mites that live on the beetle. Phoretic is a fancy name for hitchhiking mites. So they merely catch a ride on the beetle, but they are helpful. Next slide, please. They help the beetle because they eat fly eggs and small maggots before the maggots can take over this valuable resource. Interestingly, the male usually leaves a few days before the female does, and right before he leaves, these mites crawl out of the surrounding soil and cover him. And then the female has hardly any on her at this point, but right before she leaves a few days later, they crawl out of the soil to hitch a ride on her. Next slide, please. Okay, so this map, there's a lot to unpack on this map, but that green line that you see shows the former range of the beetles. It covered 35 states east of the Rockies, and those brown areas that you see there represent counties where the beetles have been found somewhat recently, although there's a few in there that probably shouldn't be there anymore. That one in Texas down there probably shouldn't be there anymore because they haven't been found there for quite some time. Now, the yellow dots are zoos that have beetle colonies and are also working on reintroduction efforts as well. So the Roger Williams Park Zoo, way over in Providence, Rhode Island, has been the one that's worked with them probably the most and longest out there. Next slide, please. So why are the beetles endangered? The truth is we really don't know, but by comparing the reintroductions that we were doing all across the US, we might find out more. One theory is habitat fragmentation. So not only does this break up the habitat where the beetle lives, but it disrupts the habitat where the animals that the beetles eat live as well. Next slide, please. Also, this fragmented habitat is really good for species that might get to the carcass first, so it favors them. So they might get to the carcass first, or they might even smell it and dig that up after the beetles had already done all that hard work there. Next slide, please. Also, many species that were right in that like perfect weight range for these beetles, like the birds that you see here on the screen are either extinct or near extinct. Next slide, please. Okay, so here's one more theory that I'll show you. So if you take that range map that I showed you earlier and overlay that on a satellite image of the US at night, now you can see that I have exaggerated that western boundary kind of to be able to show you that, but the places where it's found right now in the wild are on the edge of that artificial light glow. More research is needed in this area, but it may have something to do with it as well. Next slide, please. So what are we doing about it at the zoo? Well, we started surveying for the beetle in 2001, and you're going to hear about that in a bit. And then we started to raise the beetles at our zoo in 2004, and you're going to hear a bit about that after me. Next slide, please. So in 2005, the year after we started to raise the beetles at the zoo, we were asked by the U.S. Fish and Wildlife Service to help with a reintroduction that was ongoing in South Central Ohio at that time. And so we helped there for 7 years by both beetles, and we sent keepers there to help as well. Next slide, please. Then in 2012, we began the reintroductions here at home in our state here. So I love both these pictures so much. The one on the left shows folks from the U.S. Fish and Wildlife Service, the Nature Conservancy, and the Missouri Department of Conservation working with us to put the beetle back. And the one on the right shows the Zoo Alive teen volunteers that are also helping us with this work. Next slide, please. And now Kayla is going to tell you a bit about how we raise for them and we care for them at the zoo.
Hi, everyone. Like Bob said, my name is Kayla, and I am the Zoological Manager of Invertebrates at the St. Louis Zoo, which basically means my job is to care for and oversee all of the animals within the insectarium. Today, I'm specifically going to talk to you guys about what we do with American burying beetles. Next slide, please. So first of all, what do you even need to start breeding an endangered species? Well, you need beetles, and it's not like you can go pick up these beetles at any old place. You also need permission from the federal government. These beetles, even though we have them now, are technically not our property. They are the property of the US government. So you need to get permission. And we also needed to find a population of beetles that we felt like would be the most close genetically to what would normally occur in Missouri, so that if we were able to successfully reintroduce this beetle, we'd be able to more or less keep it as close to what would naturally be found in Missouri. So we got permission from Arkansas to go trap for 30 pairs of beetles to start our founder population. Thankfully, we weren't doing this completely from scratch because, like Bob mentioned, the Roger Williams Park Zoo has been working with the eastern population of American burying beetles for quite some time. So we were kind of able to adapt more or less a version of their protocol to fit our needs. Next slide. So, like I said, we got our beetles from Arkansas. You need to be able to breed these beetles, and you need the carrion in order to do that. You also need dirt in order to give them something to bury in. You need screening in order to keep them in the bucket, as well as to keep the soil level at the right level of moisture, basically. And you need pickles. Well, I guess more so the buckets that the pickles came in. And that's kind of how we got started initially, as we were trying to do our best to come up with a way that would be cost effective. And it so happened that at our zoo, our food service department had lots of empty pickle buckets that they no longer had a use for. So we were able to kind of utilize those in the beginning in order to both recycle those pickle buckets and keep costs low. So that's kind of what we started out is as using is those pickle buckets in order to get our beetles a place for them to breed. Next slide. So we don't just breed any beetle to any beetle. There is a program and there is a plan that we need to follow in order to ensure that we have a healthy population of these endangered species within zoos. This plan is called the Species Survival Plan. Mostly vertebrates, so anything from a rhino like their logo, snakes, et cetera, et cetera. But there are a couple invertebrates, and one of those is the American burying beetle. I mentioned Roger Williams Park Zoo earlier, and I have a picture on the right here of Lou Perotti, who is the SSP coordinator. His job is to kind of oversee all the beetles within North American zoos, and we talk about best practices for breeding and basically wanting to ensure that the population is genetically healthy. Bob, who you heard from earlier today, is the stud bookkeeper. So again, we try very hard to keep those populations genetically strong and as genetically varied as we can. So we do keep track of all the beetles that are in zoos in order to make sure that happens. There's a lot of paperwork there that I kind of pointed an arrow to because it can sometimes be a process. We've raised a lot of beetles, but thankfully we've got something that helps us out a little bit when it comes to some of that data collection. Next slide. I like to call it sort of a form of online dating. And basically, when you produce as many beetles as we have, you need an easy way to keep track of everyone because we do care for these beetles and keep them as individuals. And actually, we had a volunteer who initially helped us create a computer program that would help us keep our beetles organized. And as far as pairing beetles up, again, we try and keep them as genetically varied as possible. And we also want to make sure that we're not inadvertently picking and choosing beetles for various things. So we do a lot of random pair generating between, appropriate groups of beetles. So it's a really nifty program. It kind of sets everything up for us, and then it's able to randomly generate a pair that we know is genetically appropriate and that we know is going to be a good choice for trying to maintain those genetics in that population. And it's a part of a really cool program that does a lot of really neat things that was also later on added to and modified by one of the keepers in the insectarium. Next slide. Okay, so we kind of know genetically what is the best match, but what's even more important sometimes is making sure you have a male and a female beetle when you're going to pair them up. For American burying beetles, it's thankfully a little bit easier than it might be for some invertebrates. If you look here at this picture, you'll see that there is an orange spot above the mandibles of each of these beetles. On the left is a male, and his spot is much bigger and more square shaped compared to the female on the right, which is a little more dainty, a little more triangular. This is a really easy way for us to tell male and female beetles apart. And is a little less invasive than what we sometimes have to do because these beetles normally are big enough to be able to tell right away what is male and what is female. Sometimes they're pretty small. So there is a way to be able to look at them and look at their internal structures in order to tell who is male and who is female. But this is kind of a quick shortcut way to do that. Next slide. Okay, so we've got our beetles. We know one's a male and one's a female. Now we have to kind of set the mood a little bit. We have paired them up, and we need to make sure that they have what they need in order to reproduce. And that happens to be a dead animal. For us at the St. Louis Zoo, we use dead quail that have been raised specifically to be food for other animals. And those come into us frozen. We thaw them, we get them ready. Like Bob said, they have to be a certain size. If they're too big, the beetles have trouble burying them. If they're too small, there might not be enough food for the babies. And then it has to, you know, can't be too far gone, basically. And it also can't be alive. They're not going to kill it themselves. I have a video that I wanted to show you all, and I'll kind of explain a little bit more about the process of how these beetles work together to get their babies fed. So in this video, which we've recorded using special lights, we have shown the process over time of how this beetle pair actually will start to remove the feathers, or if this was a mammal, remove the fur from whatever carrion item they have found. These beetles have really good senses of smell, so they're able to sniff out dead animals from a pretty far distance. And you can see they kind of move it around, and they're taking all those feathers off because they want to get to the actual body of the animal in order to preserve it and feed their babies. Now, this doesn't always work out. Sometimes beetle pairs just, you know, for whatever reason, aren't a good match. But if it's a successful pair and they decide to go ahead and process this carrion in this way, then 3 days later, we'll check and make sure that that carrion item is completely buried. And usually they'll do that in that 3-day period. So you can see it's pretty much defeathered at this point. And if this video went on longer, you would see them actually burying this beetle or this carrion. So if all is successful, we come along 10 days later, pop the lid off the bucket. Next slide. And we have babies. So these adorable little Michelin creatures are American Burying Beetle larvae. Beetle larvae, or babies, look very different from adult beetles. And these ones are nice and healthy and happy and pretty close to migrating away to pupate. Bob mentioned a little bit of that process too. But eventually these larvae, you know, they're fed and cared for by their parents. They get to the point where they don't need, you know, they're at a big enough stage that they're ready to move on. And we note that to date that they migrate down. And then 45 to 60 days later, we will hopefully have adult beetles. Next slide, please. And that's what we see. So we check on these buckets every day once we get to that window and remove these beetles as soon as we see them. And then we house them and set them up in their own individual enclosures. And this is how we keep track of who is who and how we know genetically which beetles to breed to which beetles. So sometimes we end up with tons and tons of beetles, depending on how successful our pairings are. And then all of that information is entered into that program. Next slide. So when you raise a lot of beetles, you know, all insects in general don't live super long, and these beetles live, you know, less than a year. We have to keep every single beetle that does not end up going out to be reintroduced. We breed beetles generally twice a year, and some of those end up going, you know, they kind of help us get through to the summertime, which is when we release a lot of the beetles that we raise. But because these beetles are the property of the federal government, and because that's part of our permit, we have to keep every single one. So we have lots and lots of drawers of pinned American burying beetles that we keep, not just for the permit, but because potentially they can be used for scientific research down the road. Next slide, please. Caring for these beetles, like I said, we house them individually, so we have to care for them individually. All beetles are fed twice a week. Their little enclosures are also cleaned. Thankfully for us, we don't have to feed dead animals every single time. Adult beetles will actually eat live insects, and we feed them usually a combination of mealworms and waxworms. So that does make it a little bit easier on us, especially when we have so many beetles to feed and clean, especially right before reintro. Next slide, please. And we don't do it alone. So like I said, right before those reintro times, we have tons of beetles that we need to care for. And those numbers can, it can take us a while to do it, just keeper staff. So we have lots of help. We have help from lots of really dedicated volunteers who donate their time to assist us in caring for these beetles and getting them ready to go out for reintroduction. Got a lot of really eager and bright teen volunteers that do a great job helping, and then also help with reintroductions as well. And then on the right there, and the gentleman on the left in that picture, is Frank, who is the volunteer that helped us with our first American Burying Beetle computer program. And he's been helping us out at the Insectarium for quite some time. He loves talking to visitors, and he's been dedicated to helping us care for all of our American burying beetles. He also likes to change up his appearance, and it's not easy to tell, and I'm pretty sure Frank's here tonight, so I can say this, but that's not actually his real hair. He was just wearing that for fun that day. Next slide. So with that, I will turn my portion over to Renee, who's going to talk to you a little bit about what it's like to work in the field with these guys.
I found a beetle. Okay, next slide. Oh, no, yeah. Okay, so yeah, my name is Renee. I'm an invertebrate keeper at the St. Louis Zoo. And I'm just going to talk to you a little bit about our field work and what we're doing while we're outstanding in our fields. Next slide, please. So back in Bob's day, when he still had hair, and that is Bob in the right corner there, we were doing some surveys for beetles for about 10 years all throughout Missouri. So you can see he talked about where their existing range is. The black kind of areas are where they still exist. And we wanted to see if they still existed anywhere in Missouri. And you may be wondering, how do you trap a fairly nocturnal carrion-loving beetle? Next slide, please. I'll show you. So we use pitfall traps, and this involves raw chicken. We let it sit out for a day so it kind of gets ripe, and it almost looks like a jar of kombucha, and that's what it is you see there. It's actually kombucha, that's not the chicken. We have to make sure we take the lid off of it, otherwise the jar will explode. It builds up with gases. It can be pretty nasty. But once the chicken is ripe and ready to go, then we're ready to get out into the field and set these pitfall traps. So you essentially dig a hole, put a bucket in the hole, and then put the chicken in a jar with a screen lid, so that way other insects don't get into it and eat it. We put chicken wire over the entire trap because raw chicken surprisingly attracts a lot of animals, and that'll protect it from raccoons and possums. Then we put a shingle over it to keep the rain out and to kind of keep direct sun from being on it. So we'll set this up at the beginning of a survey, and then we will check the trap every morning for 5 consecutive days, sometimes a little bit fewer. Next slide, please. So this is often what we find in these buckets. It's just this writhing, noisy bucket of beetles. And we're looking for the American Burying Beetle, but sadly, all across Missouri, we didn't find any. What we did find was Nicrophorus marginatus. That's in the middle of the bottom row there is that picture. And their common name is the common burying beetle. So it makes sense that we found a lot of them. Now there are a lot of different Nicrophorus beetles. There's 15 in North America alone. So we used guides to help ID them. And after a while, you start to get a pretty good eye for it. So we did this trapping for about I think 10,000 trap nights before we decided to kind of start doing releases. Next slide, please. So it was actually ideal that we didn't find any beetles. This way we could designate them as a non-essential experimental population. And we decided to release them in Southeast Missouri for kind of a number of reasons. Bob showed the light map earlier. This is kind of a rural area, so you don't get a lot of the bright lights and busyness. It's also kind of near where beetles were last found. And we had tons of trap nights logged here already. We knew the area. Another real nice thing is we decided to release in Waconsa Prairie. That's highlighted in red at the bottom there. But there's all these other conservation areas that are managed by the MDC. And working with one organization and releasing beetles all throughout these sites is just a good plan. It's easier to work with one group. And now I say eating breakfast was important. That's because we developed relationships with the locals. So going to the Bulldog Brew in El Dorado Springs, going to McDonald's, getting coffee as we set up for surveys and mark recaptures, which I'll explain what those are. We kind of built friendships, and the people were interested in conservation and what we were doing with the beetle. It was pretty cool. Okay, next slide. So now we're ready to release some beetles. Kayla kind of talked about how this goes. So we make sure that they are male and female that we're putting together. We measure the pronotum. That's the big shield right behind their head there. We weigh them. We mark them with a medical cauterizing tool. This just leaves like a little notch in their elytra. It doesn't affect their wings or their ability to fly. It helps us when we recatch them out in the field. We know that that's a beetle that we had released and it's not a new one. And then we set the mood. So we pair them up. We don't put them in the same containers, but we get them ready to go for when we release them in the field. Okay, next slide, please. Okay, so once we're in the field, it's time to dig brood chambers. These are fairly simple. You just dig kind of a wide cap, so when you replace that dirt cap, it doesn't fall into the hole. You have a main chamber that soil and water will collect in, and then you dig a side chamber about the size of your fist. That's where the quail and the beetles go. Okay, next slide. So after you dig one, you have to repeat this a bunch of times. And this takes a lot of help from a number of different places. We have the Zoo Alive teens who are just awesome and so motivated. I love how this girl is just proudly holding 2 dead quail. It seems like we always have one. Zoo Alive stands for Active Leaders and Volunteer Education. Then we also have people from other zoos, such as Sedgwick and Tulsa. Tad from the Sophia Sachs Butterfly House is there almost every release. We involve other zoo departments, and we have the MDC and the U.S. Fish and Wildlife all there working side by side. So just to kind of explain like what's going on, in the very first picture on the left, we're marking out where the holes are to be dug. Obviously, the next is we're digging out holes. And then on the far right, we're covering it with chicken wire to prevent any possums or raccoons getting a good meal. Okay, next slide. So this year was a little bit different because of COVID. We were still able to release about 40 pairs of beetles out there, but there were only 4 of us doing it. We were wearing masks, we social distance, and we were able to get it all done in one day. It was very hot work. But we went up to Taborville this time. We're kind of walking away from Waconsa and seeing how the population does there. And we're going to start releasing in, or we have started releasing in the Taborville Prairie. Okay, next slide. Now, this is checking for larvae is something we didn't get to do this year because of COVID, but it's more for our benefit than for the beetles' benefit. Typically after release, about 10 days, we check 30% of the brood chambers to see if there's any larvae present. Now, Dan is like our larva whisperer. That's the keeper you see in the picture there. He is able to find every single last larva. It's amazing watching him like dig in there. So we check 30% and then we extrapolate what we find to the rest of the broods. Okay, next slide, please. So another thing we do is 45 to 60 days after the release, we should be expecting the mature beetles to be emerging from the broods. So we want to go see how many are coming out. On the map here, on the left, The yellow stars are all the release locations on Wakanta Prairie. The pink dots are everywhere we set those pitfall traps with the chicken. So the first day we go out there and it's kind of late afternoon, it's pretty hot, we set all the traps and then we check them for 5 days after that. The prairie often changes quite a bit when you haven't been there in a year and all your paths are gone. So we're able to load these PDF maps into a phone app called Venza, and you can see the little blue dot that's on the map on the right. That's basically where I'm standing in the prairie, and it helps direct me to the traps and makes it so much easier. Okay, next slide. So just to kind of give you a feel what it's like, the first day on the prairie is like wonderful. You see like wild mustangs like grazing in the distance. There's dick thistles and meadowlarks singing. There's scissortail flycatchers, like just went down this one road, just tons of them. And then I really like the lady tresses orchid. It's the white flower you see there. Like you just want to take pictures of every bug and plant, everything you see. But then after 5 days, it kind of changes a little bit. Next slide, please. So the reality kind of sinks in. You smell like chicken every day. The burying beetles are, they kind of release a scent on you that is not good. You have to put these little bead tags. So you can see the little green tag on the beetle. We use like a gel super glue to adhere that to their wing, and you have to be very careful, and the beetle's not very happy about it at first, but this is what lets us know that we might be catching the same beetle and we don't count something twice. And then, so along with that, we have to deal with all the ticks. Luckily, this was Bob's tick sample from his clothes. I managed not to be there that particular marker capture, so I got lucky, but they were bad. So you got to be careful with tick-borne illnesses and all that too. And I like snakes and spiders, but not everybody does. Sometimes you're going to find a snake in your bucket, or you're going to get a spider in the face. And then just from a personal story, you might be wondering why there's a pair of legs on the right there. My very first year, I wore quick dry pants, and I had no idea that my site was entirely blackberry. So I got absolutely shredded, but it was all worth it for this little beetle. So next slide. Oh yeah, and something else we do is we have field techs that are continuing to survey all 5 of these conservation areas throughout the year to see if the beetles are migrating and what their presence is. I believe we found some at Shell Osage and maybe Linscomb. I have to ask Bob that. But it really takes just some non-flinching, strong, like hardworking people to do this work every day. And I feel like I've developed friends out of all of them. They're great people, and they've moved on to some really cool careers too. Maui, Anthony is now working for the Department of Agriculture in Maui. Rachel's working at the Memphis Zoo as a keeper, so a lot of cool stuff. Okay, next slide, please. And this is where we get more into the data heavy part of it. So you may see, like with the blue bars, there is some difference there. So that's showing the number of ABBs that is released, and then it's the year on the bottom. There's some variation on what we release each year, just kind of on how successful we are at captive breeding. Now, 2013 was a year we changed how we did things in terms of releases. We had a very large storm that stalled out over the prairie that year, and we released just hundreds of beetles. And the storm dumped thousands of gallons on the prairie. So it washed out all these brood chambers that we just dug. And we were very concerned about the beetles, but that a lot of them escaped those chambers. And what we did find is they were able to find their resources and continue to breed. You can see that by the gray bar, because we were finding unnotched ABBs after that. Another kind of interesting peak was in 2016. You can see there's a lot of notched and unnotched beetles captured that year. That was just really lucky timing. We go out there and do these marker captures right when they're emerging, and we hit it just right, and the weather was great for it. So that gives us, it's very promising to see. And there is a general trend upward, as you can see by the bearing beetle line there. 2019 and 20 aren't on there, but we are, since we've walked away from Wakanta, we've seen a little bit of a dip, but we have to take into account that we've been hyperinflating the population by doing releases every year. So we can't lose hope, and we just have to kind of watch it and cross our fingers that they will continue to sustain themselves. Okay, next slide. Okay, and so I have like a little video from the field, if Emily could play that, and I'm all good. Thank you, everybody.
All right. Well, thank you so much, everybody, for putting that together. That was absolutely fascinating. And for me personally, that was definitely informative because I did not know half of the stuff that you were all talking about. So thank you for doing that. So before we get into our questions, I have a question for Renee. So obviously you're on site somewhere and it looks like there's a thunderstorm that's in behind you. So where are you at right now?
I'm actually on Wisconsin Prairie right now, live.
Oh, great. So how's the weather down there besides the thunderstorm that is by? Is it?
That's a lie. She's a liar.
The weather like here.
I brought my desk chair out here too. It's pretty hot out here.
Okay. So we have about a little under 20 minutes before we wrap up for this evening. And we have a couple of questions that have come in. So I'm going to go ahead and get us started. I'm going to spread them out throughout the three of you. So not everybody jump at the same time for the questions because I will be sure to try and spread them out as best I can. So I'm going to start with an easy one. And I think that this is a good question for all three of you. And this is a pretty generic question. So I'm going to start with Bob. I'm going to let you go first. And so the question is, what is it that made you want to get into the study and research of invertebrates? And maybe even if you'd like to share with us more specifically around the American Burying Beetle.
Yeah, that's a good question. Invertebrates. So when I first began at the zoo, people would bring in things that they would find in their backyard, and these were like amazing alien creatures. They were things that looked like they were from some film that were just like otherworldly, and so that like really took me basically. And so I often say when I worked there was not a day that I didn't learn something new, and that's honestly true. There's just so much to learn, and like I said, 98% of all animal life does not have a spine. So there's a lot to learn about them. But burying beetle is one that used to be found within our state up until 1972 or so, up until the mid 70s. So that's kind of well within my lifetime. So it was here. We could find it. You could go out, you could set up traps, you could find it in St. Louis County, you can find it in St. Louis, city. And now we can't. And so when we first began to look for it, I bet someone, I said, I'll bet you $5 that we find it. This is, beetle that lives, underground, is active only at night. You have to actively, go out and try to find it. And I lost that $5 bet because after years of looking, 10,000 trap nights, I think you said, or more, we did not find any. And that really, really made me want to find out what was going on. So it's more the fact that we're losing an entire species and we have no idea why. We don't know what's going on with it. And we can guess at certain things, but we really don't know why. And so that's I think the main sort of impetus, that these are animals that most people don't even know are out there, and we're losing them and we don't know why. So that would be my answer.
So Renee, why don't you go next? I'm going to put you on the spot.
Okay, well, I'm kind of like the hipster of the biology world. Like, I like to study the things that nobody else really does. And like insects, kind of like Bob said, seem to be like the forgotten animal. And there's still so much to learn about them, especially like in the husbandry area, and just how they exist. So I find that really interesting. I've always been kind of like, I don't know, budding naturalist. I'm always looking at the ground, trying to see something new. It's an alien world out there. And that's why I like it. And then ABBs, I just kind of stumbled onto working for the zoo. I love doing field work. That's what I did in college. So I tried to get out on every trip we did.
Okay. And Kayla?
So a little, I guess, kind of similar to both Bob and Renee. I always loved animals and always hoped that someday I would be able to work with animals. And, you know, I didn't really know what exactly I would do or with what animals I would work, but I took a really amazing introductory entomology class in college. And I realized, kind of like what Bob said, that there are so many animals out there that I don't know anything about, and that clearly, you know, there's so much to learn about them. So that kind of got me into taking more of those types of classes. I had a mentor who was a spider behavior researcher, and I worked in her lab taking care of all the research spiders, which then directly sort of put me into moving into a keeper position at the insectarium after I graduated. So it was really like just the desire to learn more about animals, which I love, and specifically about animals that I didn't know a ton about. Conservation was always something that I hoped to be involved in, and that was one of the things that really excited me about working at the St. Louis Zoo, because I read about the program with the American Burying Beetle. Didn't know that much about it at the time, but I'm like, yeah, that would be a really awesome place to work.
Oh, cool. So I have another question, and this one's for Renee, because of all the in terms of the presentation you gave around the surveying that we do.
Mm-hmm.
Whenever you all are going out and doing a survey, how many staff members and volunteers does it take to have a successful, or even just to put together a survey?
A survey? Well, shoot. So we have what, 2 or 3 people per team. So I think about like 12, somewhere between like 10 and 12 people every survey. Bob kind of was the one that coordinated them all, so he might be able to answer that a little bit better than I do off the top of my head. But yeah.
Okay. Is there a certain time of the year that you all feel it works the best? Is it normally in the springtime or the summertime? Is there a certain time of the year?
Yeah, so we release, it's usually around like May, June. And then we're checking kind of all throughout July and August, I believe.
Okay, gotcha. Okay. So then that also goes into my next question. I'm going to give this one to Bob. And in terms of the overall, in terms of the breeding program and doing these surveys and releasing some of these invertebrates that we breed at the zoo, is there other places outside of Missouri that we release these invertebrates at, or is there a certain range in which we want to keep our travel within? Is there certain areas?
So the St. Louis Zoo releases them only within our, like, so only here. And what we have set up is what's called a non-essential experimental population. So that's a government designation, and that's a 4-county area that you saw sort of carved out on that map there. And so that means that we can release them in there and that we survey in that area. Now, should the offspring of the beetles that we release there move outside that 4-county area, that county would then become part of the non-essential experimental population is what it's called. So that's happening. But I will say, so our zoo is one of 4 that are working on this. So there's the Cincinnati Zoo, which is releasing them at the Fernald area there that they have, and then there's the Wilds, which is a zoo in central Ohio, basically, that are releasing them on site. That's a large sort of reclaimed like mining area that they have, and that's like thousands and thousands of acres there that they have. And then the Roger Williams Park Zoo has been releasing them on Nantucket, island. So they've been doing that for years as well. And so all these different zoos have their own, you know, projects, and they raise them there. And we've traded beetles a little bit, but we don't like to mix the populations, you know, a whole lot, because our founder populations come from a different area, and that's a large, you know, range map there. And so the miles that are in between those small isolated, you know, populations that are out there could mean, you know, a big difference in what's called like morphotype. So you know, the size of the beetle and what they can handle
Gotcha.
out in the wild. So
okay. So switching it up, so at the beginning part of our presentation, we talked about some mites, and we did have a question that came in. Is there a signal that the beetles release that notifies the mites that they are going to be leaving the nest site? So I would imagine, Bob, this will be a good question for you.
Most likely, although more research needs to be done, I think would be the fair way to look at that. So there's something going on there, obviously, where they know that the male's getting ready to leave, and there's probably some pheromone of some sorts that allow them, you know, to know. And then the same, you know, pheromone when the female gets ready to leave. So, but, you know, a lot more work needs to be done with the mites, and there's not a whole lot of mite guys out there that do that, type of research. So it'd be good to know more about them. We just don't.
Okay. So then the next one, this is actually another good question for Renee. So during your presentation, and I believe this is during your part, you said that it was ideal that you didn't find any American burying beetles at your initial trapping sites.
Yeah.
If so, why was this ideal?
Bob kind of touched on this. This is what gave us the ability to classify them as a non-essential experimental population. So in this way, people in the community can continue their day-to-day activities and not have to worry about the beetle, like an accidental take. They could still do conservation, agriculture, recreation, all that stuff. But it did also protect the beetle from being collected or sold. So it isn't ideal that they're not in their range, but for the release itself, this was an ideal situation because we could classify them this way. I hope that makes sense.
Okay, I gotcha. So I got one more question for you, Renee, but before we get to that one, I'm gonna sneak over and I'm gonna ask Kayla a question. So in terms of the breeding program that's actually housed at the St. Louis Zoo, so in terms of the numbers that you were showing during your presentation, there were quite a few that we've successfully bred throughout the years. Does the zoo have a plan to want to expand that to be much larger than that? Or is it something that we want to continue to be consistent with as the years progress?
So I would say that where we are right now is kind of a good place as far as what our resources currently are. Like I said, we breed twice a year, and we need to get enough beetles bred so that when we breed in the fall, we have enough beetles to get us through to the spring breeding, which then leads into our reintroductions. We have 5 full-time keepers in the insectarium, and we do get help from volunteers. But we, in general, try and keep this project to a manageable degree. And like Bob said, we really do want to focus on a local, sort of on the local scale. Now, could we potentially, like Renee was saying, we've established a new release site within the last couple of years. Would we potentially move to a different site in the next few years? Possibly. But right now, we are actually trying to see how well the beetles that we've already released are doing on Waconta Prairie and then Taberville Prairie. And then we're going to kind of go from there. So we're trying to get kind of a feel for how our efforts have gone so far before we make a huge decision on whether we would expand or change anything.
Okay, awesome. Okay, so my next question is for Renee. So, and this is in terms of the actual beetles themselves. During your presentation, you talked about how whenever you're done during a survey, there's a lot of days where you go home and you smell, and it's part of the process. And so the question is, do the beetles smell bad because of their own chemistry, or is it because of the company they keep?
So they have a defensive secretion. It's just kind of this brown liquid substance, and I mean, it smells like what they eat. It smells like death. So I know when we've been taking care of them at the zoo, it's usually before lunch. So we will always wear gloves because that smell stays with you if you don't wear gloves. Yeah.
Okay. So whenever you're on site during the surveys, where is it that you all stay? Do you stay on site in a tent, RV? Do you guys go into town and stay at a hotel? Where is it that you guys stay?
So we try to keep, we want the conservation efforts to support the local community. So we have been staying at a local motel, and then we found a lodge that's really close to the release site too. So we've been kind of alternating there. But yeah, we want to try to keep it within El Dorado Springs. That's important to us, that it supports that community.
Okay, awesome. So we have about 4 minutes left. So I have one more question that I'm going to ask, and it's for all three of you. And I ask this for all of our virtual happy hour events that we do. So I'm going to start with Kayla, because I started with Bob the first time whenever we did the round table. So Kayla, the question is there a specific memory or a favorite memory that you have while you've been doing all of the work that you've been doing surrounding research or conservation surrounding invertebrates?
That's a really good question. I think I could probably come up with a few, but I think for me, I see myself in a lot of the teen volunteers that we work closely with. They're all really bright and really eager to do good. And particularly for us, we work with invertebrates, we work with beetles. I think Renee was saying, they don't really get a lot of love. So being able to, in a way, mentor a lot of these younger folks, and that includes the field techs that we are able to go out and assist for field work, has probably been the most rewarding aspect of this whole conservation effort for me. I really value the opportunity to inspire the future generation, and there's a lot of work to be done after people my age are gone and later, and it's just, that's probably been the highlight for me, is just seeing all of these wonderful young people just getting in there and working hard and being so happy holding dead quail and just wanting to do good.
Fantastic. You next. And you're on mute, so make sure you go off the mute first.
Who's that, me? Would you like me?
So
or?
Bob, you go next.
Okay. Kayla stole mine, so I'll pick another one. No, that is true, because but mine might be, so one that I recall is I was out the year after we first put the beetles out, and this was the next year. So we put them out in 2012. This was May of 2013. I was out there by myself doing surveys, and it was May, so it was early, and I found the first basically wild produced beetle that had been found in the state for years. Now we put the parents out, but this was one that had no B tags, it had no notches in it, it had nothing. And I like a slight tear went down my cheek, I'll have to say, because I was kind of proud that it worked, that this beetle, that the parents made it through the year before, and that the beetle made it all through the roughest time of year, and then I found them in late May. So that might be mine.
Okay. So we have about 1 more minute, and so I'm going to make sure that we have enough time for you, Renee. So why don't you go next?
Yeah, I mean, mine's pretty quick. So I think I just find conservation to be like awesome. And seeing all, like we go out blacklighting where we try to trap different insects, and just seeing all the diversity that's out in the prairie, and knowing that like by, you know, supporting conservation of one insect, we're kind of showing everybody that all insects are cool and helping all the species. And that's just, yeah, a brief kind of take on that.
Okay, awesome. Well, thank you all for coming to tonight's event, and thank you for Bob, Renee, and Kayla for presenting on this awesome topic. It's really always interesting to see the work that's being done through the WildCare Institute at the St. Louis Zoo, as well as the zoo in general. So thank you all for coming, and thank you for your presentation. If you want to learn more about tonight's topic, and you want to learn more about the Center for Conservation or surrounding American Burying Beetle, you can visit the St. Louis Zoo's website at stlzoo.org. We also have another event that's coming up on August 20th. So if you'd like to register for that event, you can also visit the St. Louis Zoo's website at stlzoo.org/happy hour to register for that one as well. So we hope to see you all again, and thank you all, and have a great evening.
Thank you.
Thanks, everybody.
Thank you, guys.
Bye.